All semester.
I've been trying to do it, but then you ask him, and then I'm like, dang. I've been doing it the past like few weeks.
Really?
Yeah, man.
I haven't even noticed, man. Try it again, please. Not there. Oh, it's coming.
Needs a minute.
Actually, we should be making Daniel do it. He's taller.
It's okay, I don't mind.
Oh, okay, yeah. Let me just do that.
Signal. So, I believe this is where we left off, yes?
Yep.
So, everybody is clear how we move from a double helix DNA structure to a condensed chromosome at metaphase, yes? Yes?
When you say when we
Move from, so how the DNA starts from being a double helix structure like this, and then during metaphase, we find it looking like that. That's what I'm saying. We move, how the DNA moves from being a double helix to that structure.
and it's wrapped around the histone. No wait, it's wrapped around the histone proteins all the time,
All the time.
but it's just
Only
folded.
during the times when it's being transcribed that it unwraps from the histone protein so that it can be unzipped, and then we make a copy. So
Okay,
whether it's being transcribed into another DNA, like during DNA synthesis, or when we are about to make protein and we're making RNA.
and it's put into that shape by enzymes.
Which shape?
The chromosomal folded shape,
Yes
the
and
looped
no.
domain kind of thing.
Yeah. Yes or no. It's just it associates itself with the proteins like that. Right. Wraps itself around it, forms the nucleosomes, and that gets coiled around and then gets condensed to that. All right. So then, if chromosome, even just not just the condensed, because this part, when it gets to this part, that's what we call it a chromosome. When it's not this way and it is this way, we call it what? Chromatin. All right? Yes? So when it's not condensed, so it's still wrapped around the nucleosomes. Wrapped around the histone protein, so we still has a coiled nucleosomes, but it is not considered a condensed chromosome. When it is like that in the during interphase, it looks diffuse in the nucleus, and so it's not. You don't see prominent chromosomes like you see during late prophase into metaphase. That is what we call chromatin. So then, if the chromatin is this way, all right. It needs to go through some kind of restructuring or remodeling to ensure that when we need to make copies of the DNA, we can do that. All right. So it goes through chromatin remodeling, and we do that to accommodate the DNA protein interactions. The chromatin structure must change to allow replication and gene expression. It has to be able to basically expose itself, so unwind itself from the histone proteins, and then it gets unzipped. So it needs to be relaxed. It needs to be exposed. The regions that need to be copied. Now, keep in mind also that, like for instance, when we're making proteins, the entire DNA does not necessarily have to be copied. It depends on what the cell's needs are. So if the cell needs to make a particular type of protein for a particular function, only the areas on the DNA that has the genes for those proteins gets transcribed. Right? It's just that the cell, an active cell usually has a lot of needs, so it will be actively making a lot different proteins. So you may have multiple genes being copied at the same time, but usually the protein that gets made is basically what is needed for the cell at that particular time, and then the chromatin must have a reversal mechanism for the inactivity. So once it's done, when it's inactive, it needs to be activated. All right, so the process needs to be reversed so that we can copy it, and then when it's done copying, we can go back to it being inactive again. All right. So then that also brings us to the point that basically the double helix that we have, it goes through more twists and turns, right? So based on this, you see a double helix, which is already a twisted structure, goes through more twisting and turns, right? So we form what we call the superhelix. All right. So it's the principal packaging unit of the DNA in eukaryotic nucleus is the superhelix, which is basically the twists and turns of DNA, which goes around the histone proteins. Now, one thing that we noticed, though, is that even though we have the superhelix formation, the histone proteins tend to have these tails that do not get mixed up in all the twists and turns, right? It gets left over. So these actually are the histone proteins, and this is the DNA, which is wrapped around that. But these tails that you see here, right, so you see this actually extends beyond the DNA. It is believed that it serves as points for allowing for the reversal of the inactivity to be done. So, for instance, if you need to replicate this area, let's say if you need to replicate this area, this particular tail, when an enzyme or another protein pulls on it, can relax this area so that it can be copied. That's what it's believed that the tails are doing. So the unstructured histone tails are not packed into the folded histone domains with the nucleosome. The tails are devoid of secondary structure and protrude throughout the minor groove. And so this is how it looks like, with the tails not mixed in the twist and turn. Right? So the tails, like I said, tails are believed to help in the chemical modification of the chromatin itself as well. Right? And not only does it help to relax certain areas of the DNA that needs to be copied, but it also allows for some of these chemical compositions that tends to either change or modify the functioning of a particular gene, right, or a particular DNA, or a particular chromosome. So these chemical modifications are very important to genetic function. There are three of the ones that are commonly known, especially the first one, acetylation, is the one that is commonly studied and known, and then we have methylation and then phosphorylation. If you recall, we talked about acetylation and methylation a while back. You guys remember? Don't remember, it's your homework. All right. So several of these potential, but I'm not going to collect it. Just make sure you know it for the exam.
Sure you couldn't talk about it here?
No, that was like 3 chapters ago.
What chapter exactly?
I forgot what I was saying. I was going to say something and I forgot.
Methylation.
Just like a quick review.
Equals.
No, I have to remember what I was going to say before you said all that. No, it had nothing to do with homework. Homework was a side thing.
Exam?
I'm sorry.
Exam?
No.
Oh.
No.
Oh, yeah.
Anyway, so just keep in mind that the tails are important for chemical modification. They'll come to me. All right, so let's look at these 3 different processes, chemical processes or chemical modifications that happen. So like I said, the most studied of these chemical processes is the acetylation. It involves an enzyme called acetyltransferase, or HAT, right? And what it does is that it just adds an acetyl group, right, to a positively charged amino group on side chain lysine. What is an acetyl group? Somebody done organic chemistry?
Oh, well, we should know this.
What's an acetyl group?
CH3
CH what?
I don't know.
Yeah, CH3 is methyl. That's what we're going to next.
Okay.
CH, well, not the H, but CHO.
Oh.
CHO. Right. So it adds the CHO to lysine, And then for methylation, it adds a methyl group, CH3, to either arginine or lysine. And then we have phosphorylation. Everybody knows about phosphorylation, right? Yes, no. What does phosphorylation do? Adds phosphates, right? So the enzyme that does the methylation is the methyl transferase. It transfers a methyl group from a substrate to the amino acid. The acetyl transferase. So remember, they're all acetyl transferase. Then we have methyl transferase, and then phosphorylation is a kinase. Right? Kinase enzymes add phosphates to substances. Yes, Morgan.
So what does, for acetylation, what does changing the charge of the proteins do?
We'll get there. Oh, so actually, that is going to make me talk about what I don't want to talk about. Well, adding or not adding, sometimes adding these groups, whether it's acetylation, methylation, phosphorylation, can either make the chromosome activated or not. All right. So when we talk about epistatic genes, for instance, there's when we say some of the genes are there but they're not expressed, it could be because of too many methyl groups have been added to it, so it makes the gene inactive. All right. Or too many acetyl groups have been added, it makes it inactive. And then for some, when you add it, that's when they become activated. So when it's not there, then even though the gene is there, it doesn't get turned on. Right? So that's why it looks like the genes have been masked. Well, I guess that goes to homework. Okay. So although we know that DNA of the eukaryotic chromosome consists of one continuous double helical fiber, we also know that the chromosome is not uniform from end to end. How do we know that? When you use X-ray diffraction or you use certain methods to look at the chromosome, you see that there are some areas that are lightly stained, and there's some areas that are darkly stained. So that tells you that the composition of those areas couldn't possibly not be the same, right? So when those things were noted in analysis, chemical analysis, then the scientists decided they need to figure out why is that some places are lightly stained and then some places are darkly stained. And what they found out was that the lightly stained or darkly stained is as a result of the degree of coiling, right? So when the certain areas that are highly coiled tend to be darkly stained, and then those areas that are lightly coiled tend to be lightly stained. Then they also discovered that the areas that tend to be tightly coiled are areas that have genes that we call them non-coding genes, because they basically have no effect. They are inert areas. Those are not genes they don't contain genes that get activated. They are inactivated. So they came up with the two terminology, euchromatin and heterochromatin. So the uncoiled and active areas are the euchromatin. So the more relaxed areas are the areas that you would have the genes that need to be coded for or transcribed. Those are the euchromatin areas. And the heterochromatin, these are the areas that are highly inactive. So for instance, like we know about centromere, right? You know about centromere, correct? The centromere is part of the chromosome, but does it code for anything? No. What does it do? It holds homologs together, right? So, but then it's part of the chromosome, it's part of the genome, and so that the area where the centromere is usually will be tightly coiled. So places like that. And then, like for instance, we talked about telomeres also, right? Are telomeres coding regions? No. So again, when you look at the telomeres will also show up as a heterochromatin and not a euchromatin. You had a question?
Are the centromeres made out of genetic material?
Yeah.
Oh, okay. Cool.
Okay. So the heterochromatin are basically they're genetically inactive. They don't have genes, or if they do, those genes are repressed genes, so they don't need to be activated. unless something changes and then they get activated. Also, um, it, it was noted that when it comes to synthesis of DNA or replication, the heterochromatin areas do not replicate at the same time as the euchromatin areas. It replicates a little later in the synthesis phase than the euchromatin. So basically, the euchromatin areas get synthesized first, and then when it's done, then the heterochromatin areas get replicated. So seeing this, so I gave you telomere and centromere as the two areas that are described as or examples of euchromatin. So that also showed scientists that chromosomes do not always encode proteins. Basically, what they're saying is that then it also means that even though we can see an entire length of chromosome and it may seem like it has a variety of thousands of genes, not every part of the chromosome actually contains genes that will make a protein. So it has basically non-coding regions. Now, when they look at, well, before I even say that, let me just talk about banding first. Or should I say it? Okay, I'll say it when I get to it, because then I'll get ahead of myself. All right, so basically at this point, all right, we've been able to understand how the DNA gets packaged into a cell and all that. Basically, what we can tell in terms of differences between the chromosomes are basically their sizes, right? So we can tell that, okay, this is a bigger chromosome than this one. But then when we have chromosomes of the same size, where we basically get stuck, all right? So around the 1970s, we basically could tell that, okay, this is different based on the size, and sometimes even based on the, well, even the centromere, we couldn't really tell that the centromere was there or where it's located in the chromosome. So then studies were done so that we can show different patterns. Like, remember, if you recall, when we looked at the large chromosomes, the palatine, we saw that they could actually see different bands, right, on these palatines. So then experimentation was done. Our staining techniques were utilized that showed similar banding patterns on these chromosomes, and that is what helped us to be able to really distinguish chromosomes from each other, even if they are about the same size and all that. And not just that, to also be able to karyotype, put homologs together, right? So that process of being able to differentiate the different chromosomes from each other due to the bands that they have, the locations of the genes, is basically referred to as chromosome banding. Right? So there are two different bandings that we'll talk about. Right, so the banding technique is basically the differential staining along the longitudinal axis of the mitotic chromosome. The staining patterns resemble the palatine chromosome band. So there are two types, the C and the G bands. The C bands basically indicate where the centromeres are. Right, so when you do the C staining, it shows you, so it basically helps us to see that let's say a centromere is located centrally or is located metacentrally or telocentrally, and so that's how we're able to actually also classify chromosomes based on where the centromeres are located. Right, so the only centromeres, the heterochromatin centromeres, take up that stain of the C banding, and then with the G banding basically stains the entire length of the chromosome, the P and the Q, and it shows you the different bands. So the homologs will have similar banding pattern when you do the G staining. And for the G staining, what you do basically digest the chromosome. They use an enzyme to break it up, right? And then they stain it with tyrosine. They use a tyrosine enzyme, and then they stain it with Giemsa stain. That gives a G band. So if you look this particular, I'm not sure if you can see the really dark areas. So with the C banding, the C banding shows you where the centromeres are located. So that's the only thing it stains. So It helps us. So basically, if you are going to group the chromosomes based on their centromeres, the C staining will help you do that. And then if you want to do the entire banding, so for you to be able to do an entire karyotype of a group, right, then the G banding is the way to go, because then you can actually pair up the homologs. So you see that chromosome 1, 2, 3, and even like the first 5 chromosomes, lengthwise, they're kind of about the same, right? And they're not showing the centromere location, but I can guess that parts of the centromeres, or I mean the location of the centromeres, are also not too far off, right? So then, if you're going to karyotype a cell, it's going to be difficult without the banding pattern to locate the homologs, right? So, but then the banding pattern. So if you look at this, even though this looks like it's a little bent, if you were to straighten it, it will be the same length. And then when you look at what the bands are, so there's the band here. There's another one darkened here. So it's the dark, light, dark, right? Like in the locations are about the same. If you look at it closely, you would see that the locations where the bands are about the same. Compared to this, like even I can tell the difference between this and this. This one has a dark band here at the end. This one doesn't. Right? So these banding techniques basically helped us to be able to distinguish between these chromosomes, so we can karyotype for organisms.
Is each band like a certain like?
So for each band is supposed to have genes, and you could have several. You could have hundreds of genes in a particular band.
Okay. And the one that's the lighter parts are just stuff that's not coding.
Well, there could be some, or not all of them are non-coding, right? But we will look at non-coding areas shortly. But not all of them are. All right. So, like I said, this is what happens. The banding helped us be able to distinguish chromosomes from each other. So then, after this was standardized, we were able to use these banding patterns to basically identify chromosomes. The oligonucleotide helped us to identify each chromosome. So, for instance, the X chromosome, when the X chromosome was done, so you basically see the banding pattern, right? So this side is showing us the P and the Q. Why does this arrow keep running away from? Okay, there it is. So this is the centromere. The P is the upper arm. The Q is the lower arm. Right? So then when you look at the P area, so you remember I said it's not necessarily the light areas are not coding. They could have coding. They could have genes there as well. Right? So this X chromosome, when the banding pattern was done, we were able to identify certain things. So certain locations, so P22.3, P22.2, and they basically identified the number of genes that will basically be in each of the areas. So the Q's and the P's will have different banding patterns, and that is how you're basically able to compare different chromosomes and link them or show that they are either homologs or not, even though they might be the same size but the same centromere location. So it was also noted that the areas that are the heterochromatin, the inactive areas, tend to have base pairs that are repetitive. Okay. Now, majority of, like, when you take the genome of a person, about 99% of the time, everything will be similar, right, except for the different alleles of the genes and all that. But if you're just looking at the genome, it would be similar except for about 0.1% of the genome. That 0.1% happen to be the areas where we have what we call repetitive base pair, or repetitive DNA. So we have certain base pairs that are repetitive, right? So they will repeat several times, and then the length of the repeats may differ. So even though we have the same repetitive base pairs, we don't have the same number. So for instance, if you're looking at a repetitive, a particular repetitive sequence, and I have, let's say, 10. Some of those repetitive sequences. Somebody might have 20. Somebody might have 8, and that is how. That is one of the ways that we are able to actually identify people. Right? So when they do forensics, for instance, that's how they're able to identify who was at a crime scene, right? By doing these, running a gel on these pieces of DNA that gives you the repeat. So when they take, let's say, they do it with DNA fingerprinting. So they take my sample. They take a sample from the scene, and then they take all the suspect samples. And what they do is when they run it, whoever has the same repeat as what was found at the scene has to, because everybody's repeat is going to be different. And that is actually one of the ways that identical twins get separated. Because even though they have the same of everything, when it comes to that, some of their repetitive DNA will not be the same. Right? So that's how they get separated. But for the most part, they will be exactly the same. So a repetitive DNA sequence are repeated many times within eukaryotic chromosome. Then there are different categories. Now, we will also see that there are some that we call tandem. It means that they are adjacent to each other. So you have, like, if let's say ATTC is a repeat, so you have ATTC, 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 and they are all non-coding regions, and then you find, let's say, a coding region in between, and then you get the repeat against ATTC, ATTC. So those are tandem repeats. And then you have some that are interspersed, right? And then there are some that are basically they flank coding regions, and then there are some that coding regions flank them, all right? So we'll look at the different types. So the repetitive DNA could be distinguished as, or could be identified as either highly repetitive or middle repetitive. Right? And then the highly repetitive are the ones that have been described as being satellite DNA. And then the middle repetitive ones, they are the ones that are involving tandem repeats. Right? Then with that, there are different types. We classify them as either multiple copy genes. Most genes are single copy genes, right? But there are a few that are multiple copies. Then we have the mini satellites. We have the microsatellites. The multiple copy genes are basically the RNA genes. Then the mini satellite genes, we have the variable number tandem repeats. The macrosatellites are the ones we call the short tandem repeats. Then the interspersed retrotransposons, we have the short ones versus the line ones. And examples of a sign is the ALU. The example of a line is the LY. But we'll go through all of them. Yeah, so the satellite. So these ones are the highly repetitive ones. So with short repeated sequences, they make up the variable portion of total DNA. They're found in heterochromatic central regions of chromosomes, and only eukaryotic cells have them. So basically, if you were to subject the genome of a eukaryotic cell to reassociation kinetics, you would get an centrifugation, and you get basically the absorbance and the graph of the absorbance and density. You get two profiles, right? This one, the S, is for the satellite, all right, and then the majority of the genome will be the big. So you basically have most of the genome, which is going to be almost the same across the board, and then the satellite, the repeated areas is going to be that second wave, which will be of a different density. All right, so then a couple of the heterochromatin areas, we know the centromeres. The centromeres are needed during mitotic process. What does it do? We said that it holds the homologs together, and during the mitotic process, they separate the homologs from each other. Okay? So they are necessary. They are primary constrictions along the eukaryotic chromosomes, and they allow for separation during the mitotic process. And then there are also the kinetochore proteins. These binds the spindle fibers, microtubules during the cell division. Again, this is needed during cell division. Now let's look at the moderately repetitive DNA sequences. So the moderately ones, we said we have the microsatellites and then the mini satellites. The microsatellites are the short tandem repeats, the STRs. The mini satellites are the variable number tandem repeats, that is the VNTRs. And what are they? So with these ones, they have the base sequence, repeating base sequence runs from 15 to 100 base pairs. So you could have 15 base pairs repeating, all right, or up to about 100 base pairs repeating. And they are found within and between genes. So within these, there could be flanking genes, or genes will be flanking them, right? And they will be in the middle, or they will be on the either side of it.
Are those the light colored portion of chromosomes?
No, not necessarily.
Oh.
So, like I said, the light and the dark doesn't necessarily mean it's coding or non-coding. Right? It's just the characteristic of the gene stain. So it makes some places light, so it makes some places dark. But it doesn't mean that the places that are light or dark do not have coding on them. They may have both. What actually separates, what actually helps us to see then how many of the repetitive DNA is basically when we run it through electrophoresis, gel electrophoresis, and it actually will, because then the DNA gets broken into fragments, and the sizes of the DNA will get separated when you run the gel, and then that's how you're able to count how many of the repeats you have. And in terms of their location, it may not, it's not the gene. Okay, the band will not tell you where their location is. All right. So the short tandem repeats, or the microsatellites, these are tandemly repeated. So basically, these are repeated in sequence, all right, or adjacent to each other, and they are dispersed throughout the genome. And they could range, because they are short, they could either be made up of just two nucleotides. So you could have a C, a C, a C, a C, a C, a C, a C repeating, and then you have a gene, and then you have a C, a C, a C, a C, a C, a. All right. Or you could have a CAT, 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 CAT repeat. So that is a di, tri, tetra, or a pentanucleotide. And then for the lines and the signs, the signs are short interspersed elements, and then the lines are the long interspersed elements. These ones are transposable sequences. So these are the parts. These are also, they are repeated sequences, but they can be moved. All right, they can move from one area. So the transposable sequences are the ones that are mobile and can relocate within the genome. They're dispersed throughout the genome rather than tandemly repeated. So you're not going to see them, like, even though they're repeats, you're not going to see them back to back to back. So you're not going to see the repeat, and then you see gene, and then the repeat. They're repeats, but then they are spread all over. So you can see it's here, and then you might see it like maybe downstream after so many other coded genes in between. All right, unlike the tandem repeats that are back to back to back.
But it would always be the same number of base
But it would
pairs.
be the same number of base pairs, yes.
And same order.
In the same order. Because if it's not the same order, they're not repeats.
Right.
Okay. All right. And then in terms of its composition, basically the lines and the signs make about a third of the entire genome, human genome. So it's a lot. And then there are some that transposons that they are called retrotransposons. That is because they are basically made as RNAs, and then they need to be. They're also mobile base pairs, but they are RNA that needs to be converted to DNA first before they get moved. All right. So those are, and so it's similar to when viruses, instead of having DNA as their nucleic acid, will have RNA, and then we use reverse transcriptase to reverse it back to DNA before we can insert it into a DNA. So the retrotransposons are RNA base pairs, but then they get converted to a DNA base pair, and then they can be moved like the regular transposons. So now knowing that, yes.
What are all those for?
What? The transposons, all these will help us to do DNA fingerprinting, to be able to distinguish people from each other.
Okay. What do they do in the genome?
Nothing. They're inactive.
They're chilling.
Yeah, they're just chilling. So they're inactive. They are not coding regions, but they get copied anyway because they're part of the genome. And it's a good thing they're there because that is how we're able to really tell who is who when we do these analyses.
Okay,
right?
but they essentially have no. We could get along without them.
We could, yeah. But then forensics will have a hard time also figuring out bad people who have been, or not necessarily bad people, but people who have been at certain places, or even for identification purposes. It doesn't have to be a bad thing. It could be that you need to be identified for something. You're missing. We need to identify you, and we need to know where you've been and all that. So it helps for us to be able to do that. So it basically, in studying the genetic makeup of organisms, we came to basically discover these, and we have capitalized on them and used them for our benefit. But what we know, so for now, what we know is that they are inactive areas. They don't make proteins. They don't do anything, and that's why those areas also stay highly condensed. Right. So they stay highly condensed. Because they don't need to really code for anything. Right. The only time that you would see them uncondensed is when they need to be, when there's synthesis of DNA. So those areas need to be all the areas need to be replicated. So then they will go through that process.
Right.
But in terms of activity of the cell, they are basically non-functional. All right. So basically, from what we have seen so far, what proportion of the eukaryotic genome actually encodes for functional genes? What do you think?
Not very much.
Not very much, right? Only because based on what we have learned, we've seen that there is a lot of areas that basically are non-coding areas. You could actually even say are not necessary, right? But so only a small portion of the eukaryotic genome, about 2 to 10%, constitutes the protein encoding genes. The rest of them, the large number of single copy non-coding regions, are called pseudogenes. These ones are DNA sequences that represent. So we do have these pseudogenes. These pseudogenes, basically, you can basically look at them as imperfect copies of functional genes. So it's believed that based on information that has been accumulated over the years, that these pseudogenes are basically mutated genes that have basically lost their function. So they're similar to certain genes that are working, but we don't really need these ones, but they're still there, right? So they are called pseudogenes. So these are DNA sequences representing evolutionary vestiges, have undergone significant mutational alterations like insertions and deletions, and they are not transcribed. So in addition to having repeated base sequences that are inactive, we also have these pseudogenes as well present in our genome. And then we have the ones that are necessary, which is not. So even though we have a lot, like eukaryotic cells have high amounts of genetic material, not all the genetic material is necessary. Some are, like even the non-coding regions, like the telomeres, are necessary for something. The telomeres, if they are not there, even the coded regions may not be transcribed. So the telomeres are there to protect our genome, right? The centromeres are there for a purpose also, to help with the separation, to help with to hold when cytochromatids are formed, hold them together, and then also for separation during the mitotic process. But there are a whole lot of these heterochromatin areas are unnecessary. Well, we're saying unnecessary, but I'm sure God knows they're necessary. That's why He put them there. We just haven't discovered it yet. So until we discover it, we're going to say they are not they are unnecessary. All right. So that's it for chapter 12. Well, enjoy your weekend.